Okay, we're live. So you can start.
Okay. Okay. Hi, everyone. Good morning. So my name is Meeta Mistry, and I am over on the East Coast. I'm located at the School of Public Health at Harvard, and I'm part of a bioinformatics course. So just a quick introduction to myself. What I do normally is half the time is spent consulting and analyzing data for biomedical researchers that come through the core with different projects, most of which are RNA-seq, and the other half of my time is spent training people on how to become more computationally efficient to be able to analyze their own data. And so today's session is going to be on the differential expression analysis of RNA-seq data using different tools in R. So both differential expression and R have are very close to my heart in that I've spent many years, 10 years battling with R and differential expression as well. Goes back to the days of microarray. So today we're going to be working with RNA-seq data, and just a quick overview is that what we're going to be looking at is not so much learning how to use R for RNA-seq analysis. While I will be introducing all the different tools and methods, I won't go into too much depth, but the idea is to talk about the different tools that are out there and the importance of using these tools and the underlying statistical models that help us to model this data, and also the importance of including replicates. So if you think about your RNA-seq data, for those of you who are not familiar with next-generation sequencing, essentially you take your raw reads that come from your sequencing facility, and you do some sort of quality control checks. You align them to a reference genome, and then you count the number of reads that fall within these annotated gene regions. But what you're left with is essentially a count matrix where the rows are genes, the columns correspond to samples, and the values correspond to the number of reads that are annotated or that align to that particular gene. So to look for quantitative changes in expression levels between two experimental groups, or what I will often refer to as sample classes, is not as easy as we think it is. And there are a couple of different reasons for that. So the first thing is that we're dealing with integer counts, and so they're not continuous measurements. And so back to the days when you were working with microarray data, the data is not normally distributed. So we need to use methods that can be applied to the nature of the count data. Also, replication levels with RNA-seq experiments tend to be pretty low, and partially due to the fact that sequencing costs can add up. And so generally you don't see much more than 2 or 3 replicates for a study. Of course, this is going to change if you're working with a different organism or smaller genomes. You may have more replicates. And then the final thing is there is a dependence of variance on the mean, and this is going to change with increasing number of replicates. So what do I mean by this? This is basically what I will get into more as we get deeper into the lesson. So what I'm going to ask you guys to do is, I think you all have this markdown open, and this is in the GitHub repo. So there's a couple of markdowns here, and the one we're going to be working off is called hands-on. So if you click on that, you'd be able to easily follow along with the lesson. And the first few things are just about setting up. So the first thing you're going to need is to download the data set. So if you actually click on the link in the markdown, what you'll find is that an R data object will get downloaded. Keep note of where it gets saved. For me, it goes straight into my downloads. And let me know if I'm going too fast. So the other thing okay, so now that we've got the data downloaded, we're going to open up RStudio. Okay, and what you see in RStudio is this main panel, which is the console, and then over on the right-hand side, you also have 2 additional panels, and I'll talk a little bit about them as we interact with them. So the first thing we want to do is create a new project, and we'll do that by going to File, New Project. Okay, you'll have three options. Choose New Directory and Empty Project. And I'm going to call it, give it a name, whatever you feel like is appropriate. And the important thing here about creating a project is just a note on data organization. This is a great way if you're doing a specific analysis to keep everything contained within a single project. So you can go ahead and create it. And for me, I created it in my desktop and opened a new window. Okay. So what you now should see in your bottom right-hand panel is there's basically a Finder window or your file system, and you should see a project is created, and it tells you where you are. So I'm in Home Desktop D Analysis.
Hey, Nita, could you go over this part again? I think some people, it went a little bit fast for some people.
Okay. So
downloading the information, I think.
Downloading. So the downloading is simply just clicking on the link, and it'll get downloaded. And so now what you want to do, so for me, if I open this in the Finder, it's in my downloads, and that's that file right here. And what I want to do is actually move it in into the new project that I've created. So you can either do it using your interface, like using the Finder windows, or I can show you in R how to do it, which is basically going to, so I'll go home, I'll scroll down and go into my downloads, which is where the file got downloaded. Okay, and I'm going to look for the file, which is called bottomly_eset.rdata. So if you click on that, and then you click on the wheel up here, that'll give you some pull-down options. And then you can say move. If you click on move, it'll ask you where to move it to, and you can choose the project. So now if you go back to your desktop or wherever you have the project folder, you should now see this RData object, which is in your working directory. If you're confused at all about where your working directory is, what you can do is inside the console you can type in get wd with 2 parentheses, and that'll tell you exactly where your working directory is. Okay. The second thing to do, just to get ourselves set up, is you'd want to open up a new script file or a text editor, so you can add basically a 4th panel to RStudio. And you can do that by clicking on File, New File. You have an option of many different file types. We'll click on RScript. And now up top what you should find is a text editor. And this is convenient because all the commands that we run, we can type in here and then save for later use. Okay, so I'm going to go ahead and give myself a title here and say the analysis of RNA-seq data. And whenever there's a hashtag or a pound sign, pound symbol, it's essentially indicating that this is a comment and don't run this code. I'm also going to...
Yeah. Can we hold up and make sure everyone gets the data in the right place?
Sure.
Okay, I'll let you know when we have that.
Okay, sounds good.
Okay, so if you're in the middle of this, don't put any sticky up. If you're done, could you put the blue one?
Yeah.
And if you need help, could you put the yellow one up somewhere where we can see it from the back? So right on the very top of your monitor. All right. Yes. I'll just start back here, and others about 5 or 6. So.
So you're in R, not RStudio. Did you download RStudio? So basically, RStudio is like a package for a wrapper for R. So if you're just opening R, that's what you're seeing. That is essentially just the console window that she has. So RStudio is software that has all this. Yeah, so you do R, you need R to have RStudio, but R is not RStudio. Were there instructions in the
I think there is no restriction set. Saying like there is no restriction set might be lying to me. And
First of all, you,
So that's not, yeah, that's not worried.
Okay, so the way you can figure that out.
It could be, yeah, but we're like 3 times. And the only thing that's uncomfortable is that restriction. Is it, wait,
You just go to RStudio.
yeah, I don't know. Well, I think some trigger initiatives, I think management as a unit has a large but more. It brings in members. Try this again. 3 things to do today.
so now
No, you have 4 hours and
No, but that's what I see on the microscope.
no, I don't think I have standards that I do oh my god, yeah. I really feel like anything yeah, I don't need like a couple of them. Would you say something I do like 3?
That's how I like.
You guys doing okay?
So we get the
I think that 4 too, I will. But I think it's, I feel like it's in this.
You just say, you need to figure out where this hole is.
Yeah. Like doing I think it's so you have to like be on.
Run that, yeah.
Yeah, but like
Go there, yeah. Okay, so it's in your uniform document project here. You first need to move that download there, wherever it is, you need to move it into the probably the easiest just to cut and paste it. That's why you know this space is not working, is because it's looking for the data in this folder, that's not in there. Yeah, so if you guys go, since you have the project working, if you move the data in there, it should be good. Yes. There are, okay, so we're going to use that, is that right?
1.
So, yeah.
I do, I don't want to. I have to put into a plant, another plant, bring another plant, do it, and another plant. So dumb.
So now you
Yeah, because people get used to that. Can't you do anything? Yeah, into tobacco to prove that it actually does something. When I want to be very well running, so I put them in plans to see if they do, then I know it's
Okay, so now you're going to see what happens.
it's like a better line of evidence.
You start to process.
Yeah, so that's not really working. Yeah, one of them works, but this is another one that is I want to try unfortunately,
And after that,
It's just another floating protocol with your cells, so I'm trying to use this word. For me at least.
Okay, so you're so did you start a new article? So that would probably, I would go ahead and, so it's gonna be up at the top. I think it's like a different. Yeah. So do
Do you have this?
And now I'm just going to put it to
Yeah.
Okay, so what happened is, RStudio recently
I know what to do.
had its new project,
5 micrometers.
and it also moved
Anything else?
that folder. So basically what this was is, you now have this folder on your desktop. That's where it sets all the files you can reference to be, and that's where it's going to put the files in. So all you need to do now is just find that downloaded file and just move it in there. So you can just after that, yeah. File your project. So all this is basically doing is starting a project and just another folder on your desktop. And so it's going to switch over to this project and change the world. And now what you're going to need to do is that file that you downloaded or download it again and put it in the folder that's on your desktop. Okay. So now we're going to files. Yeah, so we're just going to do it over here. And now you're ready to go. I just dragged it from her downloads into her.
Hey, Mita, we just get a couple more minutes and then we'll be ready. I'll let you know. Thank you for being patient.
No problem. Let me know if I can help explain anything. Or
Yeah, I think it's just getting the files in the right directories for places. I'm going to mute our microphone too. I didn't realize it wasn't muted.
Okay.
Hello?
Hi.
Are we, this is Lisa in San Diego. We're just waiting for those guys to load the data. Is that what's going on?
Yeah, I think so.
Okay. Okay, I had us muted and I just wanted to make sure that we were on the same page.
Okay, is everybody okay?
Yeah, we're here. I think we're good. We have the data loaded and we think I mean, everything seems fine.
Okay, cool.
There's only 3 of us, and we've used R before, so they have a harder task on their hands, I think.
Yeah, she said a couple of minutes.
Okay, yeah, that's fine. We'll just hang out and wait for them to be ready.
All right, sounds good.
Okay.
Okay, we're good to get started. We have almost 40 people, so that's
Oh,
takes
wow.
coordination. Yeah.
Okay. Yeah, if I'm going too fast, just let me know and I'll slow down. All right, so now everyone should have their RStudio looking somewhat similar to mine in that you have 4 different panels. The top panel is the script or the text editor, and that I created by going to File, New File, RScript. Okay. The second panel down here on the left is the console, and so that's where we're going to be running all of the commands. And then up here in the top right, we have the environment, and as we start to walk through the lesson, you'll see the environment start to populate with the different variables and objects that we create. And then the tab here is the history of all the commands that we run. And then the bottom right-hand corner is is a bunch of things, but the most important thing is that there's this Files tab, which is essentially your file system or your directory structure. So you should see the path to your working directory, where mine is home, desktop, D, analysis, and I have 2 things in there. One is the bottomly ESET R data object, and then an R project file. So what I've started to do is create a script, because the idea is that we're going to be running tons of commands and we want to keep track of what we're running. When you're creating a script, I highly recommend to comment liberally. Future you will thank you for that. So I put a title. You can call it whatever you'd like. And I'm also going to save this. So go back up to File, go down to Save As, and then you have can name it whatever you'd like. So I'm going to call it script.r. Save that, and now you should see down here in your file finder that the script.r now appears. Okay. So now we need to load the libraries that we're going to be using. And so I'll add a comment here to say I'm loading the libraries. And the code to load a library is basically to load the package that you should have installed prior to this class. The first one is called edjar. Okay. And so to load the library, you use the function library followed by parentheses. And then inside, you type the name of the library or the package that you want to use. You don't need quotation marks. The second one is called DSeq2. Okay, and it is case sensitive. The third one is called Limma. And I noticed that for myself that I also needed another package, and I apologize for not notifying you sooner, but it's called BioBase. So for some of you, you may have to install it, and I have the instructions on the Etherpad. So let me know, Jessica, if you want me to stop for that. The instructions are here up top, so you basically would run this command here. Source the BioC-Lite script, because you should have the bioconductor already installed, and then you can use BioC-Lite to install BioBase.
Can you go through showing up to what it should look like when it's properly loaded, and then we can get everyone to that point?
Okay. So what I'll do is highlight this chunk of code that I want to run, and I'm going to press Control-Enter. And what you should see in your console is something similar to mine. A bunch of messages telling you what R is loading. You may get warnings. Warnings are okay. If you see an error, that means that the library is not successfully loaded. Okay. All of the dependencies are also loading, and so it may take a little bit longer, maybe a couple of seconds. Another way to check that it's loaded is to use a function called session info. Okay. And if you run this function, it's basically going to tell you, and you may not have as long a list as I do, but it's going to tell you what's been loaded. And so you should see the packages that you've just loaded. So I've loaded BioBase, I've loaded DSeq, and then I also have edjar and Limma down here. So either looking at the output from when you do the library load and also the session info should tell you whether you successfully installed the package and were able to successfully load the library. Okay. Let's try to. Okay, so I'm assuming everybody's okay with that. The other thing we want to load is the actual data. Okay, and the data is contained inside what we call an R data object. And so in order to load that data, we use the load function. And this time we need to surround the name of our data file with quotations, either single or double quotations. Called bottomly. You can run that in the console. That should be relatively fast. The way to know that that data has successfully loaded is now your environment should have populated. So this data object basically contains 3 large expression sets. Expression set is basically an object that contains the expression data, so the actual count matrix, along with any kind of phenotype data, along with any kind of metadata having to do with the samples, and also data having to do with the genes themselves. Okay, so if you don't see these 3, and if you don't see the session info stuff, definitely ask for some help for that.
Okay, we're going to take another minute, I think, to get this working, and I will let you know when we're all set.
Okay, sounds good.
Thanks.
I have a question actually while we're waiting. So how do you create expression set file types. So like when we have our data coming into it, usually it's in tabular form.
Yeah.
Yeah.
So sorry, I'm hearing a. If you don't mind just your side again.
Sure.
Thanks. Okay, so the way to create a new object is using the biobase package, and you would, I think the function is eset maybe. I can't remember exactly. I think it's something you create new and then followed by eset. I can't remember the exact commands, but it's using functions that are available in the biobase package.
Okay.
I think at break I can pull up the code for that.
Okay, yeah, I guess. So I've used mostly EdgeR before, and what I've put into it is just kind of like the table format of my, counts, and I haven't converted it. But so is that your original input? Is that what you use, and then create an expression set from that?
Exactly. So all you need is your original any kind of metadata, feature data, if you have any, about anything like annotating your genes. You don't need to have an expression set object. The only way reason I've set it up like this is for the purpose of distributing this data set. But usually I don't always work with expression sets.
Okay, cool. Thank you.
No problem.
Okay, sorry about that. Let's keep going.
Was there problems with the installation?
Yeah, there's just different dependencies are not working on some people's computers, but it's working for most people, so we can just go ahead.
Okay. Okay, so once we have the data and the libraries loaded, we can go back to the Markdown. Okay. So let me just introduce the data set and where I got it from and why we're using it. So when you look inside RStudio and you see these 3 expression set objects, they correspond to 3 data sets that I've created using this original bottomly data set that's available on Recount. And so Recount is this. If you click on the link, it's an awesome open resource provided by Johns Hopkins School of Public Health, where they have a bunch of data sets here for RNA-seq. They're publicly available. They have their published studies, so they have the original publication, and then they have links to the expression set, the count table, and then the phenotype table, which is the metadata. Okay, so that's where I got it from. And the reason there's 3 is that the original data set contains 2 different mouse strains. So they're looking at C57 black and the DBA strain, each with 10 and 11 replicants respectively. So what I did was I randomly subsetted this data set, and so I created a smaller one with only 5 replicants, and then I created another one with only 2 replicants for each strain. Because I wanted to demonstrate how the relationship between mean and variance will change as we increase the number of replicates. So when I'm talking about expression set, if you wanted to see what exactly is contained in here, you can just type into your console bottomly 3 reps and press return. And so a bunch of information gets spitted out to screen. So it tells you here that you have your assay data, which is essentially your count matrix, has 10,228 features, which correspond to genes, and then 4 samples, right? And so for pheno data, this is just your metadata, so any kind of information that you have on each of your samples. And feature data is the ensemble IDs. Here, some people also have official gene symbols, for example. So from now on, for here on in, I'm going to start copy-pasting just in the interest of time. But I'll try to explain where I can the different functions that I'm using. So the first thing I want to look at or explore is the relationship between mean and variance. And to do that, I'm going to do it for each data set separately. So I'm going to copy this first chunk of code up here for the 2 replicate data set, and I'm going to paste it into my script. Okay. So the first step here is assigning the 2 replicate data set to a variable called eset. Okay, and now you'll see that show up in your environment. The second one is creating a CPM matrix. So it's basically normalizing the data by sequence depth, and it's taking the log transform of that. So now you have, if you look here, this is a large matrix of values. Okay, where your rows are your ensemble IDs and your columns are your sample IDs. Now I'm going to create a vector that computes the mean. So basically this is going to go, this function here will go through the CPM matrix and go row by row and calculate a mean, an average expression level for each gene. The next line here is going to do the same thing, except rather than computing the mean, you're going to compute the standard deviation. And this final line is going to actually plot this data. So what you should see in the bottom right-hand panel is, and this is problematic because it's too small, so I'll run it again. I wonder if I can open that. There you go. So this is probably closer to what you see on your own. And so you have standard deviation on the y-axis, you have the average log CPM on the x-axis, and what you see is this sort of scatter on the low end, and that scatter tends to be reduced as you get to larger values of expression. Okay. Before we talk a little bit about this, I want to also go ahead and plot the 5-replicate data set. So if you go back to the Markdown and you copy the next chunk of code which corresponds to the 5-replicate data set. Okay. And then you can paste it into your editor and either run each line separately or highlight the entire chunk and then press Control Enter. And now what you see is a similar plot, but what you see is the scatter has been slightly reduced on the low end over here. Okay. Now let's take a look at the last replicate, which is the 10-replicate data set or the full data set. Okay. So again, I'm going to copy that and then paste it into my editor. I'm going to highlight that whole chunk of code, Control Enter, and now you'll see yet another plot here. Okay. And so compared to the 5 and the 2 replicates, you see this reduction in the scatter as you increase the number of samples that you have per sample class. So when you're working with RNA-seq data, genes with larger average expression are going to have, on average, larger observed variance across samples. So what that means is that there's the genes that have higher or more highly expressed are going to vary in expression from sample to sample more than the genes that tend to have lower expression values. This phenomena is what we, or having this different scatter across the range of expression is known as heteroskedasticity. And so that's essentially what we're observing in the figures that we just plotted. The thing is, because we took the log transform, this counteracts this and actually reverses the adjustment. So basically, now instead of seeing that scatter on the high end, we've basically seen the scatter on the low end of expression. Okay, the scatter that we see is, again, as I mentioned, as we increase the number of replicates, the scatter tends to get smaller or is reduced. And so the standard deviations of averages are smaller than standard deviations of an individual observation. And so if you think of multiple observations, so fewer versus more observations, the standard deviation is going to shrink furthermore. So if you're adding more data or more replicates to your sample class or your experimental group, what you're going to get is you're going to increase the estimates of your group means, and so with more precise estimates, you're going to be able to more easily be able to tell apart or tease apart the means which are close together and identify whether or not they actually are statistically significantly different from one another. Okay, and so that's what those, and that's essentially showing you how the replicates are affecting the mean variance relationship and explaining what the mean variance relationship has to do with the end product, which is identifying differentially expressed genes. Okay. So how do we best model this type of data? Knowing that there is heteroskedasticity, or there is this, we tend to see this scatter with RNA-seq data, especially when working with smaller sample sizes. Well, because we have such small sample sizes or low number of replicates, distribution-free, rank, or permutation-based methods can't work. The Poisson model was what was originally proposed when RNA-seq data analysis first started. The problem is that it's a single parameter model with mean is equal to variance, and as you've seen from our real data, we actually have more variance than the Poisson can explain. Right. So more recently, statistical models have turned to what the statistical tools for RNA-seq analysis have now turned to the negative binomial model, which is a good approximation of the Poisson, but it has an added parameter which models the dispersion, or basically the within-group variation. So how much variation, given you have 2, 3, 4, 5 replicates, how much variation is within group, and how is that compared to your between-group comparison? EdgeR and DE-seq2 are the two very popular tools which implement differential expression analysis based on this negative binomial model. The two offer very similar functionality, but they do differ in the way that the dispersions are estimated. And they do perform very similarly too, as you will see. So let's start with DE-seq2. Both DE-seq2 and EdgeR utilize this concept of borrowing information from genes. So rather than computing a gene-wise dispersion parameter independent of every other gene, use the whole data set to compute a common value or a trend. And then you shrink these individual gene-wise estimates towards this anchor. So with DE-seq2, the first round of estimates are going to be computed using maximum likelihood. And so once you have an estimate for for dispersion for every single gene in your matrix, a curve is going to be fit. And then once that curve is fit, the gene-wise estimates are shrunken towards the values predicted by the curve. And we'll see that as we start to plot these mean dispersion plots. So the first step in using DE-seq2 is to first create what is called a DE-seq2 dataset object, and that's the first line. That's the first line in this code, but unfortunately it is cut off. So let's see if I can fix that on the fly. Sorry, this will help. This will help in copy pasting and reduce errors. All right, so scroll back down. There we go. So essentially, what we're doing is specifying the first item, which is the count data. And that's the expression data inside our expression set object. You could just as easily add your count matrix here. You do not need to use an expression set object. I simply created these for ease of use of these data sets, and also the third tool that I'm using does use expression set objects. Okay, so what you can do is copy that first line there, or first two lines. Let me copy that whole chunk. Okay, so I'm going to copy the whole chunk of code under create DE-seq2 object datasets. I'm going to paste it in. Okay. And again, we're specifying the expression, so the count matrix. The next thing is defining where our metadata is kept, and that's also within our expression set object. And then the design is the formula to indicate what is the factor that we hope to find expression differences within. So let's take a look. Let's do a quick look at what's inside our expression set object. I'm going to clear this. So we passed in expression of the bottom lead 2 reps, right? Or we passed in the bottom lead.eset. Okay, and what I'm going to do is wrap this inside the head function, so we only see the first few lines, but at least you can see what we're working with, right? So what we have is just our count matrix. We have ensemble IDs for the genes on the far left, and then for every column we have a header which corresponds to the sample name that we're evaluating. And then within there we have the count values. Okay, so we can do the same thing, except now we can look at the P data. And the P data is essentially the phenotype data or the metadata. So now what you have is each row corresponds to the sample name that you saw in the count matrix, and then you have information. So you have the strain, the experiment number, and also the lane number that it was run on. Okay, so this is the information that we're passing to this function called DSeq dataset from matrix. Okay, and by running that function, let's see if we can. Okay, so by running that function, what we'll see is that now we've created an object called DDS, which is a large DSeq dataset object. And if you click on that arrow, you'll see a whole bunch of information. So it's basically created this object that can hold a whole bunch of information, and as you go through the different steps of differential expression analysis, that information will get stored in a slot that is designated for that information. So at the moment, the only things full in these slots are the data that we've provided, which is the count matrix and the metadata and the design formula. Now to create or to generate more information and populate more slots within that object, we run the next line. Okay? And that's the DSeq function, which is doing a number of things. And it actually is quite verbose. So if you watch in your console, you should be getting a lot of information spit out to you. So the first thing it's doing is normalizing. It's estimating size factors and doing a normalization. Then it's estimating these dispersion factors for each gene, right? And then there's this gene-wise dispersion. You can ignore the warnings for now. This is something that we won't talk about in this course, but it's fine to move forward. The important thing is to look at what it's doing. So it's the dispersion estimates, and again, the final dispersion estimates, looking at the mean dispersion relationship, and basically doing the fitting, the model, and the testing, and essentially looking for differential expression. So everything has happened, and all of the results are now stored inside this object. And we'll get to more of these results in a little bit, but let's go ahead and first run or create these data sets for the remaining 2 data sets that we're working with. Okay. So I'm going to copy paste the remaining code for the DSeq section here. Okay. I'm going to put a note here saying this is the 5 replicate. I'll run that. And then I'll put another comment here to specify this is the 2 replicate. Okay, so I'll run the last one, which is the 2 replicate. Okay. And so once you're finished, you should see your command prompt or this greater than symbol back in your console, which means it's ready to go. Additionally, you should see in your environment the 3 objects have been created. DDS, which is your full data set, DDS.2rep, which is your 2 replicate DSeq object, and DDS.5rep is your 5 replicate DSeq object. The next thing we can do is use the information, because remember there's dispersion parameters stored within those objects. We can use the information stored in those objects to plot dispersion estimate against mean plots. All right, so let's take that first one. Actually, let's copy paste the whole chunk into our editor. And then we'll run them one at a time. Great. So this is the plot that was generated for the full data set. And the black dots represent the first pass of gene dispersion estimates. The red line is the fitted curve that is based on the original black dots there. And then the blue dots are the final dispersion estimates. Okay? And so you see that the blue dots in this 10 replicate data set do not deviate so much from the original estimates, which is the black dots, for the most part. Okay? So now let's take a look once we reduce the number of replicates to 5. So here you see there's a little more shrinkage happening in that the blue are closer to the red line, and there's a little bit of deviation from the black dots. So now let's try the final data set, which is the smallest of the replicates. It's 2. Okay, and here now you see that a lot of the black is showing, indicating that the original estimates have been severely shrunk towards this fit. Okay, so what this means is that as you decrease the number of samples that you're working with, the shrinkage is going to increase in strength, right? And so you saw that with the 2 replicate data set, meaning that for many genes, when you're shrinking it that much, when the shrinkage strength is so high, you're actually overestimating, which in the end may actually lower the rate of true detection when it comes to differential expression. Okay. So that's, again, demonstrating the mean-variance relationship, but in the context of one of the tools that we're using, which is DESeq2. Now what we'll try is look at the same sort of thing, but using EdgeR. So EdgeR is actually similar, again, that it's borrowing information across genes, except the difference here is that a common dispersion is first estimated for all the tags, and then an empirical Bayes strategy is applied for squeezing the tag-wise dispersion towards that common dispersion. The difference here is that it's the amount of shrinkage is actually determined by a prior weight, also considered as a prior degrees of freedom, as opposed to the DESeq2, where those dispersion estimates are shrunk based on the fit to the initial estimates of dispersion. Okay. With EdgeR, the biological coefficient of variation, also called the BCV, is the variation with which the unknown true abundance of the gene varies between the replicate samples. So basically, how much variation is there within group? Okay, and dispersion estimates are obtained by taking this BCV value and squaring that value. So what we can do with EdgeR, rather than creating these, rather than having this function to generate the plots that we had, we're going to have to we're going to be basically looking at the BCV values, okay, the squared BCV values. So in order to run EdgeR, we're going to have to do something similar to what we did with DA-seq2, in that we have to create an object. Again, it's going to be, instead of a DA-seq object, this time it's an EdgeR object, also called DGE, Differential Gene Expression object. And what we're going to do here is, again, specify the same information. It's just a different sort of syntax. So here you're going to specify the counts is equal to the count data that's stored inside your expression set object. And then the second parameter is group. Group is corresponding to which factor inside your metadata is the factor that you wish to evaluate. Okay, so we'll copy paste the first two lines there. Okay, I'm going to create a new section called B with EdgeR. And run that first line of code. And take a quick look inside my environment. So here is DGE. Right, and again, you see not as many because, again, the two tools work differently, but you have these slots, and some of these slots are empty, and some of these slots are occupied with information that we have provided. Okay, so the next line of code is calculating these normalization factors. EdgeR has a slightly different method for normalization than DA-seq2. DA-seq2 uses a median of ratios method, whereas EdgeR uses a TMM normalization. I don't have time to go into the details, but they differ ever so slightly, and if you actually plotted them against one another, the correlations are quite high. Okay, so this function basically calculated normalization factors, which is based on sequencing depth, and stored those values into our object. Okay. And then I'm going to copy the next few steps here. The next part is to create what we call a contrast matrix, and this is basically specifying the samples that we want to compare to one another. So let's actually run those two lines of code. Okay. And then in the console, I'm just going to type in design.mat and press return, so you can see what these contrasts are exactly. So what you have is every row corresponds to a sample in your data, in your original data set, and then every column corresponds to the different factor values. So we specified that our group is strain, and we know that strain has two categories. So a single sample can only be one of two categories, C57 black or DBA. And so as we go through each column, if we go through each row here, that means that's corresponding to every sample, and it's telling us or identifying which class or which group that sample belongs to. So we know that sample 1, 2, all the way to 10 belong to the C57 group. And then we know that the remaining samples, because of the 1, so it's a 0, 1, it's a binary matrix, 0 indicating not part of it, 1 indicating part of that. And so we basically created this matrix to let edgeR know which samples we want to compare to one another. So we've let edgeR know that 1 to 10 are contained within one group, and dispersion estimates can be computed based on the fact that we have 10 samples within a single group, followed by another 11 samples in the second group. Okay. So now that we have all of the required information, we can go ahead and estimate dispersion parameters for edgeR. And these are slightly different functions than DESeq, but essentially what it requires is the DGE object, which we created, followed by your design matrix, which we also created. The first step is just to create a common dispersion of essentially getting an average across the all genes. The second step is computing a trended dispersion, and that's again sort of creating that curve similar to DESeq2. You can actually skip the common step and go straight to the trended dispersion, and from the trended dispersion you can also compute what we call tag-wise, and tags refer to genes, so it's estimating per gene dispersion parameter. You may find that this takes a little bit of time.
Can I ask a quick question while we're waiting on this?
Sure.
So when you're saying, so you're running the trended off of the common because you've renamed it DGE, but you were also saying that you could just run line 68 on its own and it would automatically call common first?
I don't know that I'm not sure if it calls common, but that's a good question. So one thing to clarify is that by specifying CD on the left-hand side, it means that I'm just adding information into that object. It's not to say that I'm overriding the data. So yeah, I do think that by calling trended on its own, common should have been called through trended, but I'm not entirely sure how that works.
Okay, but that answers my question. So in this format, when you're renaming DGE, you're not replacing it, you're just adding to the object.
Yeah, so it's very interesting because normally using R, if you were to do that, you'd actually over.
Okay, yeah, that helps me out a lot. I've seen this code before and that totally confused me.
Yeah.
So that's really helpful. Thank you.
No problem. And the same goes for DESeq2. When you up here, we just ran a single function, but there are actually separate functions that are being called within that function. And so if you wanted to run it step by step, you would do the exact same thing by just going through and adding it, using this notation to fill in slots inside this object. Okay, so it looks like we have all of our dispersion information now compiled within our object, which is actually this one here, DGE. Okay. And now we can go ahead and do this all over again for the two remaining data sets. So this first chunk here is for the 5-replicate data set. I'll copy paste that. Okay, so I'll run that in. This time I'm creating a new name because I want to create a separate object altogether for the 5-replicate data set. And again, I've created a design matrix which indicates which samples belong to which strain. Okay, let's run these last 3 lines, which are basically computing the dispersion estimates. Okay, and while we're waiting for that, I'll jump back to the Markdown and copy that last chunk of code, which corresponds to this 2-replicate data set. So I'll also copy that tiny little bit of code at the bottom here, which is plotting of the BCV. So the BCV value can be interpreted as the percentage of variation that's observed. And so we'll look at the values that we get for variation when you have larger number of replicates versus a smaller number of replicates, and how that changes by plotting these. Okay, so first, now that the 5-replicate has been completed for me, I'm going to run that first chunk for the 2-replicate data set. And then I'm going to run the last few lines to compute dispersion estimates. And while that's running, I'll also just mention something about the model.matrix here that we've done, or creating the design matrix. We have the tilde, and then we have a 0 plus. You could also run this with a model where you don't have this 0, and essentially what you would have is there's no intercept, and you're just comparing one to another. And so you'd only have one column indicating whether it's DBA or not DBA, or C57 or not C57, because you know what the baseline is. It would be, I believe it does it alphabetically, so it would take C57 to be your reference level. All right, so it looks like we have all of the objects created for each of our data sets. So let's go ahead and start with the full data set. I'm going to increase this so it doesn't give me an error. Okay. And so what you see here is, not sure why it's doing that. So what you see here is the blue is the trended, and the red is the common, which is the average, which in this case is very close to 0.2. Maybe 0.19 is the value for the full data set. And you can plot that for the 5-replicate. So this is, I'm not sure why I didn't see this on my my full data set. You're supposed to see these black dots, which are the original estimates for each of the genes. So each dot here, similar to the DEC plot, represents a gene. And then you have the trended curve that's being drawn here, and then you have the red common trend, which is being drawn here. Okay, and again, you can see both with this plot and the DEC2 plots, you see that scatter and the scatter reduction, although you saw it maybe to a lesser extent, but you do see the reduction of that scatter as you have increased the number of replicates. So let's take a look at that last plot, which is the 2 replicates and the BCV plot. So again, you have your average log CPM, which is your mean expression value, plotted against your coefficient of variation. So they're just different metrics of showing the variance, right? And so what you're plotting against, plotting here is the mean against the variance, and all of these tools have a function where they're internally looking at these, or assessing the heteroscedasticity And these are things that you should look into too when you're working with very tiny data sets. Usually, if you're on the range of 2 or 3 data sets, this usually isn't too problematic. But if you're seeing a lot of within group variation and the amount of between group variation that you're looking for is very subtle, that signal is very small, then it's going to be very hard to deduce signal from noise. Okay, so I'm not sure if you guys want to take a quick break before we start on the next section, which is the next tool, or keep plugging away. I guess for both.
We're fine to keep going.
Okay. Jessica, are you guys okay to keep going? Okay, so it looks like we'll take a quick break.
So if you need to take a break, that's great. I had a couple quick questions that maybe I could ask in the meantime, when you get back, if you are going to take a break.
No, I'm fine.
So that was really helpful. I think kind of to follow up, so one of my questions has kind of been to then understand the differences between the packages and to know kind of which ones to use in different situations. And so these examples kind of seem like they're giving similar results. Have you found that with your stuff, or I'm just curious what you've settled on? Or
so they're very small in terms of the result. What we have found is that DESeq2 is a little bit more stringent in that you'll get fewer genes. But if you look at the overlap, there's generally high overlap. We, at the core, we have a personal preference for DESeq2, and we're kind of biased because Mike Love is one of the developers and also here at the School of Public Health. And so whenever we need help with anything, we can bug him. But I think both perform equally well. I've never seen a paper where the two are not very concordant. So it's just a personal preference, to be honest, with those two tools.
And what about with Limma?
So Limma is, and I'll talk about the transformation that it does, but essentially that's something that we use when we have either what we think is really noisy data and it's hard to extract signal from there. So if you were to do like a PCA or clustering and you didn't see your data sort of segregating, it was just kind of all natural biological variation that you're seeing. Or alternatively, when you have a lot of different factors to consider, because what Limma is doing is it's basically the Voom transform is it's computing some sort of weight which is applied to your original count data, which transforms your data into now normally distributed data. And now that it's normally distributed, you have access to a lot of these mature linear modeling tools that were used for microarray data. And so when you have a lot of different covariates that you're working with, these tools can come in really handy.
Sorry, there's a delay on my unmuting the mic every time. Yeah, that makes sense, because something that, a lot of the experiments that we have two factors that are a matrix, and it's something we've talked about using EdgeR with generalized linear models using essentially the negative binomial versus Limma. And I guess I've been unclear. I understand the difference in the models and converting to normalizing, but just like functionally, is there any reason why using the edge R models does it not perform as well? Or like, let's just say if you have two factors, or does it kind of just depend on the data set?
Yeah, I think it's more data set dependent. I haven't played around with it much. I've just in cases where again, like I see that it's very noisy and I'm unable to find anything with DSeq2 or EdgeR. I'll try basically using the Voom transform. There's really, unfortunately, there's no gold standard for the field, and there's also no rules saying, use this in this situation and use this in another situation. So, but these are definitely.
Yeah, that's kind of where we're at too, is figuring out if there is a goal and realizing that there isn't, but then understanding the pros and cons so we know what to use when. And I think that's where a lot of us kind of are still figuring it out. So yeah, talking about it is helpful for sure with people.
Yeah, so that's been data that's come more, and so that's what we've decided on basically. Those are our sort of rules, in-house rules, that if it's noisy or if we have a lot of covariates that we want to model, then we stick to Voom versus DSeq2.
Okay, cool. And in the edge R with the different dispersion, so everything I've read and used is kind of always using the tag-wise. Is that what you guys typically? I mean, this is what we have here, but I'm just curious if that's what you typically do.
Yeah, you definitely do the tag-wise because the tag-wise gene-wise, and you need gene-wise dispersion estimates. I've not had a situation where I didn't use the tag-wise, but I guess there may be examples in the manual. I haven't explored it as much.
Yeah, there's a little bit, but it seemed to be the consensus was that, so I was just checking.
Yeah.
Okay, cool. Those were all my questions. Thank you.
Okay, so I'll address these two questions that are in the Etherpad. So the first is, can you discuss the difference between CPM and TPM? So counts per million is basically just normalizing by sequencing depth, where TPM is more of a proportion of transcripts that are, and I can't remember the exact computation or the exact.
I'm going to just pause because Jessica was typing that they're not back yet.
Oh, okay.
Sorry. I think because their mic's out, so she can't say anything.
Okay.
She says we're almost there.
I just saw that. Sorry.
Oh no, I didn't notice it either.
All right, so I'll get started again. I think before I head back to the material, I'll answer some of the questions that are in the Etherpad. So the first question that came up was, can you discuss the difference between CPM and TPM? And so when it comes to normalization metrics, there are tons of them out there. The idea is that you're basically trying to get your count data scaled so that you're making a fair comparison between samples, between genes. CPM is simply taking your count and normalizing it by the total number of reads, so your sequencing depth for that sample. With TPM, it's actually what they call a measurement of the proportion of transcripts. And so I don't know the exact formula for that, but I know that it measures the rate, so the number of counts per base. And so the idea is that it's telling you, given a specific, the TPM is the number of transcripts that you would have seen for that transcript given the abundances of other transcripts in your population. But again, I don't know much more detail about that. And again, DESeq2 has its own normalization, which is a median of ratios method, which is very well explained in the DESeq2 paper, which basically creates a fictive reference. So it'll create a new sample, which is a fictive sample based on expression values in your entire matrix, and then compute a bunch of ratios, and then using the distribution of ratios, take the median value as your scaling factor. FPKM and RPKM, I'm sure many of you are familiar with. That basically takes into account sequencing length and also gene lengths. Okay, so the next question is, can we also discuss what we need to consider for input data so we can apply it to our work? And that's a great question. So essentially what you need for input is a count matrix and a metadata matrix, or a metadata table. And that was something that I looked at the beginning, so maybe I can pull that up again. Okay. And so the count matrix is stored right now in our expression object, but you don't need it to be stored in an expression object. I'm going to say data is the name of the variable, and then I'm going to extract the values out of it. And you guys don't have to do this with me. You can just watch. And so the head function, or rather I could use the view function on data. Okay, and that's going to pop up here in my editor. And so this is what your data looks like, right? It's just a matrix. The first column is your row names, whatever you want it to be. So in this case, our data original annotation file was from Ensemble, therefore we have Ensemble IDs for every row. If you used UCSC as your annotation, these will be different. And then you have up here sample names. So each column corresponds to a different sample. Okay, and then the value inside each cell is a count value. So how many reads map to that gene? So how many 369 reads mapped to this transcript in this sample? Okay, so that's your data. That's the first thing that you need. And then the second thing you need is metadata. Okay, and this is stored again inside here, but you don't need to use this expression set object. You can simply provide them as is. So let's do a view on meta. Okay, and so this is what you'd need to provide for metadata. The bare minimum is having one column with all the with all your sample names, which correspond to the column headers in your data matrix. And then you need one column here to specify the experimental group that you're interested in comparing. So for example, this would be at minimum what you need, this column, where you basically have, for each sample, you're identifying which strain it belongs to, and because that is the comparison that you're interested in. There are other things here. These are mostly metadata having to do with the experiment, so what lane it was run on when you did the actual sequencing. You can include as much or as little information as you want, as long as you have the bare minimum, which is your column names and your factor of interest, your biological factor of interest. Okay, so that's the important thing also to remember, is that when you're when you have the two data sets, make sure a quick sanity, actually not a sanity check, you should do this before you start any kind of analyses. Make sure that the order of your samples as they are listed in your data matrix are identical to the order as they are listed in your metadata table, because otherwise that will cause you problems later down the road. Okay, there's another question about can you explain the contrast again? And for example, what is each column estimating, and what part of the script is making the comparison between the groups? Okay, so the contrast, so let's look at the design matrix. Okay, is basically specifying for each sample which sample class it belongs to. And we know that this group or this factor is what we're interested in, because when we gave the design formula, we said tilde and we specified strain, right? Or in the case of EdgeR, we created a variable called group and we specified group, which is essentially strain. And so now what we have to do is specify to the tool that we're using, so EdgeR or RNA-seq, to say which sample belongs to which sample class. Okay, so in our case, we have two sample classes, or two experimental groups, which is C57 and DBA, and it's just a binary matrix to specify. So if we had sample 5, for example, if we had another sample that belonged to the C57 group, we would have an additional row with a number 5, followed by the number 1, followed by number 0. 1 indicating it's part of this class, 0 indicating it's not part of this class. And so this matrix itself does nothing on its own, but it's useful when the tool is doing the generalized linear modeling. So now the tool knows which samples to compare when you're fitting the data to the model. So it's saying, okay, these two samples, the expression from these two samples correspond to C57 expression, and these two samples, 3 and 4, correspond to DBA expression. Okay. Hopefully that makes a little more sense. What is the difference when you exclude the 0? When you're excluding the 0, you're plotting an intercept, but more simply, what is it doing in terms of the experiment? You're basically taking out this column altogether. So you've said that this is your baseline. So now you're evaluating expression changes relative to your baseline. So C57 is your baseline, and you're saying for each gene evaluate expression and see whether it goes up in the remaining samples compared to C57 or goes down in the remaining samples compared to C57. That's the sort of the best I have for explaining that. I will admit that I am not a statistician. I'm mostly self-taught when it comes to stats. I'm really just interested in this stuff, and so I dig around in the vignettes and the manuals and the papers to find out as much information as I can, but that's about all I have for that. For this last question, plotting the dispersions, what are the values with near zero dispersion? Those are genes that are basically not changing within group or between group. So these are genes that are pretty much constitutively expressed. They're not changing at all, and so that's what the near zero dispersion is reflecting, I believe. Although oops, sorry, that's wrong. Sorry. Okay, I think I'm going to move on in the interest of time back to the material. Hopefully I answered your questions reasonably well. If you have other questions, please feel free to email me. I'll put it on the Etherpad and we can discuss later. Or I can take questions once we get through maybe a little bit more material. Okay. So I do want to get to at the very least the last tool, which is Limma-Voom. And so the previous two methods are negative binomial methods, and they treat dispersion estimates as if they were known parameters, accounting for some uncertainty without actually accounting for any uncertainty estimation. And so they can tend to increase the number of false positives that you see. What we found in the core is that we find actually more false positives generated with EdgeR than DSeq2. DSeq2 tends to be a little more stringent, but both methods are, again, they don't account for any uncertainty of the estimation of those parameters, and so this can be problematic. So we're basically now introduce a different tool, so it doesn't use the negative binomial distribution, but it also doesn't use a Poisson distribution. It actually goes back to the normal distribution, which is what you used to use for microarray data. And how can we do this? Well, basically what Limma-Voom does is it has a function which is Voom stands for variance modeling at the observational level. It's a transformation that's applied to your read counts. So it uses the variance of your genes and then creates these weights that are applied to the original count values, and now transforming your once count matrix into a new count matrix, which is basically now reflects data that it fits a normal distribution. Okay, so these 4 steps describe how this is done. So each gene-wise linear models are fitted to the normalized log CPM values. And when you fit it, you create a residual standard deviation for each gene. So then you compile all these standard deviations, and you fit a robust trend to those values. Okay, and then you take the inverse square predicted standard deviation for each observation, and that essentially becomes your weight for that observation. And when we say observation, we mean count value. Okay, so you're just to simplify it, you're taking the variance from the genes to create weights and transform your count data into another expression matrix, which is now normally distributed, and instead of using generalized linear models, we can use linear models. When is it usually best to work with Limma-Voom? So I've actually only fiddled with it very few times, and so this is, again, a lot of these tools are personal preference. So Limma-Voom for us, or at least my personal preference, is to work with it when I'm working with generally noisy data. Or the other situation that it's great for is when you're dealing with a large number of factors. And so if you have a lot of covariates, if you have to model interactions, it's really useful to use mature linear models that we've been using for a long time as opposed to generalized linear models. And so you can use these linear models because the data is now fitting to a normal distribution. Okay. So the first chunk of code here is, again, creating a design matrix. Okay, so we'll copy paste the first maybe 3 or 4 lines of code, and it seems like it's cut off again, so I'm just going to quickly fix that. I apologize. GitHub used to do this wrapping thing. I guess it's not doing that anymore. So now you should be able to easily copy paste. And again, we'll go through this line by line. So we'll call this section Limma-Voom. And we'll run that first line, which is creating, again, a design matrix similar to the one that we had before. And let's take a look at this design matrix. Okay, so here you have the case where, which was actually described in the question, what happens when you don't have that 0? What happens when you don't have that 0 is you now have this situation. As opposed to having a column here that represents C57, you now have a column for the intercept, which is 1, and you have only a single column for your factor, or your factor value, or your sample class, which is called DBA. And that goes to say that the 0 indicates that this sample number 1, is not part of the DBA class. And then when you get down to sample number 11 and onwards, you now have the value 1, which indicates that sample is part of the DBA sample class. Okay? So because we're using linear models, I believe, so I've always done it the opposite way, where I don't plot the intercept when I'm working with generalized linear models. I'm not sure that it does or doesn't work, so I don't want to give false information. With Limma, you want to do it this way. The next step is calculating these normalization factors, and you do that using the calc norm factors. Okay, and we're going to save it to a variable called NF. So again, this is just based on sequencing depth, so the total number of reads per sample. And we can take a look at what's inside NF, right? So these are our normalization or our scaling factors. You'll notice that this function looks quite similar to what was used in EdgeR. That's because Limma and EdgeR are actually created or developed by the same person. And so they share some functionality. Okay, and then this next line here is basically doing the Voom transformation. So it's taking as input your expression data, which is your count matrix. It's taking your design matrix, so the contrast that we just set up. It's taking your library size, which is basically the column sums, so the total number of reads for each of our samples multiplied by that scaling factor that we just computed, okay, which is NF. And there's a different scaling factor here for each individual sample. There's different methods to normalize by. We're going to use quantile normalization, and then we set an argument plot equals true, so a plot will be generated. So we can go ahead and highlight that and run it. Okay, let me expand this so we can see a little bit better. All right, so this is just a similar plot to what we've been plotting all along. The only thing is this time the expression level that's plotted on the x-axis is not your CPM. It's just taking the count value, it's adding 0.5, which is basically to help with the fact that if you have 0 counts when you do a log transform, you don't get NaN values. So so basically you have the log2 transformed values of expression on the x-axis, and then on the y-axis you have the square root of the standard deviation. So we're just looking at the same trends again. You see it's very tight with not a lot of scatter because we're working with the full data set here, and you can evaluate how that changes as we go through with the different data sets. Okay, so I'm going to copy that last chunk of code there. So let's do the same thing for the 5-replicate data set, which is creating the contrasts, computing the normalization factors, and doing a Voom transform. Okay, and this is the new plot. Okay, and then we'll do the final thing for the 2-rep data set and run that. And you see here with the 2 replicates, we see quite a bit of increase in that scatter on the low end of expression, which if we didn't log transform, we would see the scatter on this end over here. All right. So now that we've explored the 3 different types of 3 different tools and how to basically load data in and do the initial sort of normalization, now we're going to go forward with the actual differential gene expression analysis and then evaluate how many genes are identified with a 10-replicate data set versus a 5-replicate data set versus a 2-replicate data set, and also see how the genes overlap between the tools. Okay, so for differential expression, we're going to set a threshold of FDR less than 0.05, which is pretty standard, pretty standardly used. It's also fairly liberal, so basically you're saying there's a 5% chance of a false positive occurring within your data. And so let's set that up and store it as a variable so that we don't have to write out 0.05 every single time. So I'm going to just create a new heading for differential gene expression analysis using different tools. I'm going to put a comment to say set threshold. And then I'm going to run that. So now that value of 0.05 is stored. Let's start with edgeR. So the DGE object that we previously created contains the original count data. It contains our metadata, which is information on our samples. It contains normalization factors and also dispersion estimates. So now what we need to do is take all of that information together and use it for each gene to fit the expression data to the model. Then once the data is fit to the model, we then use the contrast to specify the comparison we make. And the comparisons are basically, the contrasts are used to let edgeR know which coefficients to use when you're making the comparison. Okay? So the question that we're asking is how many genes are differentially expressed between C57 and DBA? The first thing that we need to do is specify our design matrix. Okay. And again, we know that our DGA $samples $group is, let's just double check that. Yep, so this is for the full data set. Okay. We have basically indicating which samples belong to which group. That's what this vector is that we've added. So we're specifying that in our model, and that's going to create this design matrix. Which we've seen a few times now, which is just a binary matrix indicating which samples belong to which class. Okay. The second line is adding column names, so it's easier to refer to the different columns. Now we'll go back to the markdown and copy the rest of the code, and then we'll go through it line by line. Once we've created the contrast, the design matrix, set up our contrast, now we can do the actual model fitting. And we'll do that by using this function, which is called glm fit. The input required for glm fit is your DGE object, which contains all that information that we've already compiled, in addition to the contrasts. Okay, so what is fit? It's a large object with a lot of different information in it. So we could try typing in, sorry if you guys missed that. I use the function names, okay? And then in the parentheses, I put the name of the object that stores all the information after doing my model fitting, because I want to know what's stored inside this object. And so you see there's a lot of information that's stored, a lot of statistics that are stored. So you have your coefficients, you have your fitted values, your deviance, the method that you use, your counts, your unshrunk coefficients, the residuals. So all of this information should you need it. What we're going to do is actually use this function to put our contrasts in the appropriate format so that we can do a differential expression analysis using this GLM LRT method. Okay? So I'm going to take a look at the contrast that I've generated here. Oops. Sorry, that's contrast. Okay, so it's the same. It's basically taking our original design matrix and condensing it into this format, which is specifying that we want to find genes that are upregulated and genes that are downregulated. So upregulated in C57 and downregulated. So we were looking for both up and downregulated genes. So I'm actually a little confused about this because I think you can use either one of these two methods, but let's go ahead with the code here. So edge our results. To get the results, we have to access the information that's in the fit. So let's run this LRT method. Okay, and then we'll access the results tables from that LRT object. And let's take a look at what's inside EdgeR objects. So this is the data, these are the results that are generated based on the contrast that we specified, saying that we want genes that are either up or down regulated, and also specifying the the data itself with the normalization factors and the dispersion estimates. So the first column here is the log fold change. You have your ensemble IDs, which correspond to the different genes that you're evaluating. Log fold change followed by the log CPM. So this is the expression across all of the samples in the data set. A log odds ratio, and then an associated p-value. So at this point, nothing has been multiple test corrected. This is just the p-value that's generated based on the test statistic for the comparison between C57 versus DBA. Okay. Here, this next test, or this next function, decide test DG, is used for to specify a threshold at which you would like to say identify genes as significant versus not significant, and so that's where we've entered p.threshold. We also want to correct for multiple testing because we're doing thousands and thousands of independent tests. So the method that we're going to use as a commonly used one is the Benjamini-Hochberg method. Okay, and so this will give us our significant genes. And if you want to take a look at what's inside the significant genes, it basically returns to you a vector that tells you 0, it's not differentially expressed, versus -1, which it's negatively expressed, versus a +1, which is upregulated. So up is positive, down is -1, and 0 is not. So this line of code is saying, give me the row names of edgeR results, okay? Because if you noticed in there, it only gave us a vector of values saying 1, 0, or -1. But we didn't have any gene names. But we know it follows the same order as this matrix up here. So what this line of code is saying, give me the row names from this edgeR results table at which the values in this vector here are not equal to 0. So as long as it's not equal to 0, it's either 1 or -1, which means it's either up or down regulated. Okay? So if you press Control Return, and then we take a look at what's inside genes.edgeR, what you'll see is the ensemble IDs for the genes that are identified as significant based on edgeR. Okay, well, let's move on to DSeq2. Just double check if there's any questions here. Okay. So for DSeq2, similar to edgeR, we have that object, right? The difference is that when we ran the DSeq function, it did everything for us in one fell swoop. So under the hood, there were a lot of individual functions that were being called, similar to the way that we had to do it for edgeR. DSeq2, that actual function DSeq, just ran them all for us. And when you saw that it was pretty verbose and that it told us at every step what it was doing. So it did all the statistics from normalizations to generating coefficients to actually doing the model fitting. And now all we need to do is specify the comparisons that we need to make and extract the relevant data. Okay, so the format for specifying contrast for DSeq2 is creating a list. So there's another way of doing this as well, but I'm not going to go into that. But know that there's two methods. Okay, so let's copy paste this piece of code here, and let's call this DSeq2. Okay, so the first line here is creating the contrast, which essentially you need to specify in a list form the name of the coefficients that you want to compare. And how are you going to know this offhand? You're not. You need to check that. To do that, you need to use a function in DESeq2 which is called results names. And inside you put the name of your DESeq dataset object. Okay? And that's going to return to you the name of the different coefficients. So you have your intercept, then you have your C57, which is specified this way because what DESeq does by default is it'll take your factor, so this is your actual factor, which is strain, and then it'll append it to whatever the classes are. So strain C57, and then you have strain DBA2J. This is important because you need these exact names when you're specifying your list. Okay? So when we run that, contrast.DESeq, you see what we've generated is a list. It's slightly different from a vector. It's a different type of object in R, and you have one element of the list versus the second element of the list, and within each element is a different coefficient that's stored. So now to create a results table for the specified contrast, we use the results function. And here we specified the DESeq dataset object followed by the contrast. Okay. DESeq also has a nice function, I believe it's called summary. So if we put our results inside there, it'll summarize the information that is stored in our results file. Okay, so what this is telling us is that at an adjusted p-value of 0.1, we have 929 genes that are upregulated, 783 genes that are downregulated, 0 genes removed for outliers, and 0 for low count. So there's several thresholds. There's 3 different thresholds actually that are applied to DESeq by default, and they specify how many of these genes were removed due to these filters. And in our data, we see nothing, which is good. Okay. The other thing that you can do with the summary function is you can specify. So I believe you can say, let's see. So if you ever are stuck in R, you use the question mark followed by the function, and then in the right-hand panel here, you'll get help information. Okay, and so this will tell you how to use the function. And so there is also a way of specifying. So if you say alpha summary and then you say alpha, and it was pretty liberal, the threshold they had was a p-adjusted of 0.1, and we know that ours is going to be 0.05, so we can check how those results change if we change that threshold. Okay, and so by being a little bit more stringent, we reduce the number of differentially expressed genes. Now we have 729 and 579 that are downregulated. There are tons of other things that you can change in here. I do encourage you to explore the user manual. Okay, but for now, let's go on to what we're going to do is move on to the next line of code, which is basically setting up in our results table. So let's take a look what's inside the results table that is generated from DESeq versus EdgeR. Okay, so this is DESeq2 results. So we have a little bit more information that we saw with EdgeR. So our first column here is our row names, which are our genes. Then we have a base mean, which is the average log expression for average normalized expression for that gene across the entire data set. This is the log 2 fold change, log fold change standard error, the actual statistic, the p-value, and then the adjusted p-value for each gene. Okay, so what we want to do now is add an additional column that is called threshold, and inside that column we're going to have true and false based on whether or not our gene passes the threshold, which is a p-adjusted or a multiple test corrected value of 0.05 or an FTR of 0.05. Okay, and that's exactly what we're doing here. So DSeq results is our table. Then we're going to say $ threshold. That's the notation that we're going to use to create a new column called threshold. Okay. And as.logical means that we're creating a logical vector, and this is the threshold that we specified earlier. So now let's take a look. Scroll right over to the side. And so you see now we have another additional column that's been added on to our matrix or a data frame, and we have a value for true if it passes or false if it doesn't pass. Okay? This is just going to help us filter our data a little bit later. So now we want the genes or the ensemble IDs that correspond to the rows which have a true in the threshold column. And so that's what this line is doing. And so you see, if you go into your environment, and we close this up, you should see now a variable that's been created called genes, right? Genes.DSeq. Right here, and it tells you the total number. You could also use the length function. Okay, so it tells you that there's 1,300 genes. And now we'll do differential expression with our last tool, which is Limma-Voom. So I'll copy paste that. Okay. Again, we're going to create a design matrix. That's the first step. The usual Limma pipeline. And this is, when I say usual, this is the Limma pipeline that's used when you are working with microarray data. Okay, so it's linear modeling. LMFit is modeling the data according to the design matrix that you've specified. And then it does empirical based estimation. Okay. And to obtain the results, you use the function top table. Here you need to specify again the coefficient that you are interested in. We know that the first is the intercept, and so obviously the second is the comparison. Adjust equals BH, which is Benjamini-Hochberg. And how many genes do you want? Do you want just the top 50 genes, the top 100 genes? We actually want all of them, and so this is what that line of code is specifying. We want all of the genes irrespective of threshold. We want the full results table. And we're going to view what's inside the Voom results table. Okay, and so you have, again, your ensemble IDs followed by log fold change. You have average expression values, your test statistic, your p-value, your adjusted p-value, and not entirely sure what b is. Okay, so that's what's output from Voom. Again, what we're interested in is the adjusted p-value and genes that meet our threshold. So we're going to use the same line of code which we used with the DSeq data. And that's just going to add an additional column to Voom results. Okay, so if we take a look and we scroll over to the very end, we see now a new column that's called threshold. True for values are for genes which are identified as significant based on an FDR of 0.05, and false if they're not. Okay. Now that we have that, we can isolate the genes based on their ensemble IDs. And we're going to use a Venn diagram to depict the overlaps. We can also just take a quick look at how many genes are identified with each. So let's do a quick check. How many genes are significant? And we could, again, use that length function. So length followed by genes of Voom, length of genes.dseq, length of genes.hr. And if we just open up this, so we see the largest number of genes is actually found with the dseq2 method. Similar numbers are between Voom and hr, but how do they actually overlap with one another? To do that, we're going to need to install another package. So in the interest of time, I might ask that you, if you want to follow along, you're more than welcome to, but in case you run into problems, maybe just follow along on the screen. I actually have this library loaded, or I should. Okay, and this is really a most basic of Venn diagrams. There are other packages out there that make much fancier plots. This is just to sort of quickly give you an overview of how these genes overlap with one another. Okay, sure. Okay, so I'm just going to open this and run that again. Okay, so there was a question that came up like regarding which tool do you use, which is the best. Honestly, there is no gold standard. These are probably the top in terms of if you look at citations. They're very popularly used and they compare very well with one another. So you see that's a pretty good overlap between the three of them. 790 genes. There are a few genes identified uniquely to dseq2 and hr, but there is in general a consensus across the three different tools. So this is a good sign. I mean, this tells you that you have a reasonable data set that you're working with. And you'll see how this changes if you continue with this is using the full data set. So if you were to use the data set with 5 replicates versus the one with 2 replicates, what you'll start to notice is that you'll be picking up more noise than signal. And so here you're more certain that what you're finding with each tool is in fact true signal because of the fact that you have enough replicates to estimate that variation across within sample. Okay, so we're getting really close to 3 o'clock. I'm not sure that we can get through all this. Just check in with Jessica. Oh, okay, I just noticed that there's some unanswered questions. Can you show us how to output the differential expression data from hr with the following columns? Yes. So the data is contained in hr results. So in order to write that to file, you would use write.table. Okay, that's the function that you're using. Now you need to specify the object, which is, I think it's called hr results. Okay. Following that, you have to specify where you want to put this file, or basically give it a name. And so you can call it hr results.txt. you need to separate, identify a separator, so it can be comma or tab delimited. In this case, I put tab. Something I also normally do is I put call.names is equal to NA, because otherwise by default R will shift all the headers over one, so that you actually don't have a header for the first column, but it'll now contain the column from the second. That's the header from the second column. And then I also add in quote equals false. This essentially will make sure there's no quotations around all of the values. So if you do that and you go to Files and you scroll down, you should see an EdgeR results, and it'll open up here. And so you see that this parameter here, put a tab here as opposed to everything moving over. Okay, so it's tab delimited. You should be able to open that with Excel or whatever program it is that you're comfortable with. Okay. Oh, you've got answers. I just realized that you had some questions answered. I see you don't have the FDR p-value. There is definitely a function that will allow you to... Okay. All right, I'm going to move down to the unanswered section, which is, does upregulated here mean that the gene is more highly expressed in the first member of the contrast or the second? Okay, that's a good question. So the contrast, the up versus down is relative to whatever you've defined as your base level. And I'm not sure how this works with EdgeR, but with DESeq2 it's alphabetical. And so if you look at the fold changes in DESeq2, they should be a positive means that it's upregulated in dBA versus, if it's upregulated, it's higher in dBA relative to C57. If it's downregulated, it's down in C57. Sorry, it's down in dBA compared to C57. So let me just take a look at DESeq2 results. Okay, so for example, you have, these are pretty small fold changes. Let's take the largest one that we have here that's positive. Okay, we'll take that. And then we can plot the we can plot the expression, which is expression at bottomly set. So we'll look for the expression for that gene. It's kind of hard to tell, but usually what you can do is if your samples are labeled with something more meaningful, you can take a look at whether the expression data is actually higher in one versus lower in the other. But I know for DESeq, at the very least, for EdgeR, you'd probably have to look through the manual a little bit. And I think Limma is the same. You basically have a base that's determined based on alphabetical order. If you want, you can use something like, there's a function called relevel, which basically reorders the levels of a factor. And again, you'll have to read a little bit more about factors and how they're exactly, how they work, how this type of R data structure works. Could we use the code DESeq2 results DDS contrast alpha equals p dot threshold in order to make DESeq2 call differentially expressed according to our 0.05? Yes, so you can definitely do that. That's a great point. So the question is, can you just subset this data instead of doing line 155 here, just do that in using the summary function. I don't think you can do that using. Yes, so you can actually, not the summary function, but within the results function. So if you look at the help manual for results, there are actually many different parameters that you can provide. And so you can say here, you can specify a log fold change threshold. Right now it's at 0, but you can say, in addition to an alpha cutoff, an alpha is your p-adjusted cutoff. So here you could specify 0.05, which is what we had. In addition to that, sometimes people add a fold change threshold, so you can add that here, and then you would also just get a subsetted table, as opposed to the way we did it. And so I can demonstrate that here real quick. So you would have new results, let's call it. We would basically do results, PDS, contrast equals contrast.dseq2. Okay, and then here we would do alpha is equal to 0.05. Okay, and we could use the dim function, to find the dimensions of our new results. So this is how many genes are in our subsetted data, and now in our original DSeq. Oh, that's interesting. So let's take a look at what's inside it and what it actually did. I mean, that is essentially what it's supposed to do, but I'm not entirely sure why the dimensions are the exact same. So I'm not entirely sure why that doesn't work. I wonder if summary has anything to do with it. So if we do a summary, you still have. Yeah, I apologize. I'm not entirely sure why that doesn't work. That is supposed to. You're supposed to be able to basically specify these parameters, and then your data should be filtered. In the interest of time, I'm going to move to the next question, which is, are there ways to incorporate more metadata information, i.e. other parameters, to classify sets of replicates, other conditions, and other parameters? You can add in your metadata, I'm not sure if I'm understanding this question correctly, but you can add as much or as little as you want in your metadata table. That's the best way to add information. Especially, it'll come in especially handy when you are doing downstream visualization. So the more columns you add to the data, to the metadata table, is basically more information that you have on your samples. So you could have replicate information. You could have, for example, if these strains had different genotypes, you could have genotype information. You could have all sorts of stuff, and then you could also model that using these tools. I think there is a limit to some extent, but I'm not entirely sure. The next question is, can you suggest any sources? I can definitely add some more resources to my Markdown. and papers that look at comparisons between these methods and also papers that have used these methods. I will definitely add something at the bottom here in terms of resources to the Markdown. Okay, it looks like we're done. Thanks to everyone for joining, and I hope it wasn't too confusing. I will definitely update the Markdown with resources and anything else that I think is appropriate. I think the EdgeR stuff got a little confusing, so I'll try and fix that up a little bit, add more information there. That Markdown will always be available, so feel free to go through it on your own. And I'll also add my name up here, my email up here. If you guys have any questions and you want to email me directly, you're welcome to. Again, I'm by no means no expert in the field, but I'm happy to answer questions best I can. Okay, thank you.